I'm excited to announce our keynote speaker for the afternoon. Anne Carpenter is an institute scientist and Merkin Fellow at the Broad Institute of Harvard and MIT. Her research group develops algorithms and strategies for large-scale experiments involving images. The team's open source cell profiler software is used by thousands of biologists worldwide, including here at the Allen Institute for Cell Science. And that's available at cellprofiler.org. Anne is a pioneer in image-based profiling, the extraction of rich, unbiased information from images for a number of important applications in drug discovery and functional genomics. Her presentation is titled, Using Images as Data to Accelerate Drug Discovery. It's nice to see you again, Anne.
Thank you. Thanks, Greg. It's really delightful to be here together with this community that was started now 14 years ago by Han Chuan-Peng, and seeing so many familiar faces and so many new ones that are coming into this field. It's really delightful. So the mission of my laboratory is to bridge the gap between the latest computational methods and everyday biologists who want to get things done. And so many of the new developments that many of you in this room are making, I hope we can help be a part of getting those into the hands of biologists and then ultimately applying them to a lot of different projects that make a real difference in the world. So I just want to begin with something you're probably familiar with, Moore's Law. I think this is just an incredible triumph of human technology. The fact that the costs of computing have decreased so dramatically over time, but you may not be familiar with Eroom's law, and that is the pharma industry's observation that discovering new medicines is really hard, and it's getting harder over time, or at least it's getting more expensive. So this is a log scale, so if you had $1 billion 30 years ago, or sorry, 50 years ago, 70 years ago, you could get 30 new drugs onto the market. And as you may be aware, $1 billion doesn't even get you a single medicine these days with all the research and development and clinical trials that goes into it. So this is a field that's quite ripe for disruption, and I think computation can play a role in this. And part of my mission is to try to figure out, are there any clever things that we can do with images that can make a dent in this rather depressing plot? So today I want to overview, of course this is not just going to be the work from my own laboratory. I really want to give you a picture of how the world has been changing in terms of how images are used in biology. This includes work from many laboratories that we collaborate with and many of you in this community, but this revolution has taken us from a world where a biologist comes to us and says, oh, I really want to measure this thing. Can you help me measure this thing? And then we struggle and figure out how to do the segmentation, or maybe we don't need to segment. Maybe we can figure out how to measure what they want to measure in some other way. Maybe it uses machine learning, maybe it doesn't. But fundamentally, there's a class of experiments where the biologist knows what they want to measure, and they come to a bioimage analyst for help in measuring it. We'll start there, but I want to quickly move to using images more as data in the sense of profiling. And so we'll talk more about that as we get to it. So let's start at the kind of more simple end of the spectrum of just measuring what we know we want to measure. And many of the experiments that we work on in my laboratory are high throughput experiments. My lab is dry, but the laboratories we collaborate with are taking care of the sample preparation and designing some fancy biological assays to address some particular problems. And because we work in this high throughput space where we have thousands or tens or hundreds of thousands of images in an experiment, we tend to be working on the kinds of algorithms that can be made very robust, and by the time we're through with them, they tend to be very useful for a lot of a variety of other people's experiments as well. And so that's what led us to create Cell Profiler over time. This is open source software. I wrote the original version in MATLAB starting in 2003, but it's since been rewritten by an actual software engineer into Python and has been available for quite a while now. So in a typical experiment, you've got the sample preparation, whatever cells or organisms go into the plates, and then whatever treatments, whether they be drugs or genetic perturbations. You carry out the microscopy, you do some kind of segmentation, and then you measure everything that you can measure. And as I said, typically a biologist is is like, that's fantastic that you measure 1,000 things from my cells, but I really only care about this one thing. And so many of the experiments we've done have been of this form, and I won't dive into any one of these in detail, but just to give you a visual sense of the kinds of things biologists often want to quantify in the course of studying a particular disease. Some examples are shown here. Some of them are very low resolution, where we're looking at entire organisms, like C. elegans growing in 96 well plates. Some are very high resolution. This is a nucleus that's been irradiated in a cancer study to look at the DNA damage process, and so on. Lots of different examples are shown here. You can kind of just get a visual sense of, sometimes they're measuring how much the intensity or the amount of a certain kind of stain. Sometimes it's the morphology of cells. Sometimes it's a fancy system like a tissue sample. Usually that's not so high throughput, and so on. And sometimes they're time-lapse or three-dimensional imagery, which adds its own complications with tracking and with, as we've seen in many of the talks today. This work has had translational impact, and the 3 projects on the right are those that we worked on, where in one case on the left, it led to a successful clinical trial for a form of leukemia. The other 2 have found drugs that were useful in animal models and could progress in that direction. But I'm really proud of the projects on the right, where people just downloaded our software, used them in experiments, in the top 2 cases to identify potentially useful drugs, one of which is heading to clinical trials. And in the bottom case, actually using Cell Profiler in a clinical trial to guide patient treatment. So this kind of image analysis is definitely something that can make a difference in the real world, and especially in the context of the pharma industry, which I'm not showing here because they tend not to publish those papers. But it really is making a difference, making these physiologically interesting biological systems and then using imaging as the readout for them. So what is next in this bin of projects where the biologist knows what they want to measure? I would say that in general, researchers who are working on biomedical applications are most interested in recapitulating the disease in a dish in as physiologically relevant a manner as possible. And we've seen some examples of this in some of the talks so far, where that means ideally it would be a tissue, it could be an entire organ, there's organoids and spheroids, which have some 3D structures that are more realistic than a 2D cultured cell line, and so on. And some examples are shown here from some of the projects that we've worked on in this general direction. So I think when we talk about, there's so many things that are not challenging anymore in bioimage analysis. If somebody just has a mammalian cell type that's a cultured cell line that's just plated in a dish, it's usually pretty straightforward to segment those images very easily and automatically, and especially with deep learning. But some things kind of push the edge, and of course, that's where a lot of you are working. We are in the middle of a revolution, as you're aware of, in terms of deep learning being applicable to image analysis. And so my personal vision here is that I fully expect that in the next, let's say, 2 to 5 years, whenever a biologist buys a microscope, it comes with some software to operate the microscope. And I hope that that software knows what nuclei look like and knows what cell boundaries look like. And in just the same way that your photo software knows the members of your family and can pick them out usually without any tweaking these days, we hope that it will be the case that models have been trained on a diversity of images to the point where they approximate a biologist's brain looking at an image and can, you don't have to tell me what stain you've used or what cell type you've used. If you just say, here's an image, can you point out the nuclei? That's a thing that a biologist can do. And we're hoping that we can train models that have that kind of capability so that a lot of the tweaking that is currently necessary for configuring image analysis algorithms becomes a thing of the past. So we're working in one, I really want to make a pitch to everyone. As I said, my mission, I don't care who does what, I just want to make sure that biologists' needs are met with the latest technologies. And so we really want to serve, my lab really wants to serve in a kind of community building role to bring together researchers that are working in different areas and just make sure that each of these different pieces pieces is in place. And so for deep learning especially, it's such a dramatically different technology in terms of the sometimes the hardware that's required, the libraries that are required, and the way that you interact with a potential software that serves this field, that there's a lot of pieces that need to be built nicely for an ecosystem to work that will help biologists use deep learning. And so I do hope that if this is an area that you're interested in, I'd love to hear from you and get in touch with you to talk about how the community can work together to build some of these necessary tools that we need. Certainly my lab is not planning to build everything here, but we would love to be helpful in coordinating the community to come together on a lot of these different kinds of tools. And I've been in the field long enough that if enough people get together at the beginning and kind of coalesce around a strategy that they think will be useful, it can produce software that makes a difference because there's a critical mass of effort being put on concentrated in one. In contrast, when everybody builds their own thing totally independently, you end up with 15 things that don't really serve anybody's needs, but they get published and then die shortly thereafter. So I'd love to see this be an opportunity for the community to work together on something that can be really great and useful to the field. One way we've been using crowdsourcing towards this overall goal, specifically the goal of creating a trained model that knows what nuclei look like across imaging modalities and across staining types and so on. Some examples are shown here. Is we organized the 2018 Data Science Bowl through Kaggle, and I should say that the slides are available, and you'll see the link at the end of the talk as well. So if you can't see some of the citations here or whatever, you're welcome to take a picture of the slides, but you can also download the slides afterwards. So in this Kaggle competition, we painstakingly annotated a lot of nuclei, and so that was almost 30,000 nuclei that myself included and our whole lab had expert biologists working on. And what this nucleated was a community of researchers in mostly deep learning that ended up solving this problem very well. Now, some of the solutions are shown here. Some of the solutions were not so practical. So the winner was an ensemble method of 32 different trained networks that the implementation would be kind of horrific if you tried to make a nice tool out of that. But those that were close behind, and I would call functionally equivalent in terms of their accuracy and performance, were actually pretty straightforward and easy to implement. And so as a reference here, we have kind of a cell profiler tuned pipeline. Not necessarily, that's not like the absolute best that anybody could do with classical image processing. It's a set of pipelines that were created in like a 3-hour time period, so as quick as you could. And so what this tells you is that because the testing here was on image sets that were completely, and image types and cell types and stains that were completely not included in the training set, what this tells you is that if you had a choice between having an expert sit down with some classical methods and spend a couple of hours trying to work out a method versus just running it through any of these trained models and not touching a thing and no parameters whatsoever, those trained models would do better, which I think is really exciting. So of course there's some limitations at the edges. We didn't include bright field imagery in this experiment. We thought that would be a little too ambitious to start out with. But it's pretty exciting to me, and I'd really love to extend this towards finding cell boundaries and also to bright field. Would love for it to be 3D. This was a 2D challenge. But the fact that a pre-trained model, if you just give it a random new image set that it's never seen before, that it can do better than a person with a few hours configuring algorithms, that to me is pretty exciting. Speaking of machine learning and transitions from classical to deep learning, you might be familiar with one of our tools called Cell Profiler Analyst that's now more than 10 years old. And it's a staple in a lot of biology labs because it's a really simple interface to just drag and drop and classify different phenotypes that are found in images. And we've used that tool to screen all kinds of more complicated things. Instead of just measuring one feature, we're measuring a phenotype that uses a lot of visual cues in order to make decisions about the phenotype. And some examples are shown here. And in this space, as I said, the community is already coming together, and this is really exciting. So Peter Horvath's lab started working on advanced cell classifier around the time my lab started working on cell profiler analyst, and we didn't find out about each other until we were 2 or 3 years into the development of both tools. They have a lot of overlapping capabilities. They have a few interesting features that don't overlap that make them kind of distinctive, but it really was a shame because if we had known about each other's work, we could have worked together on a single thing. So we're not making that mistake again. Now that we know each other exist and we're both interested in the space, we're creating a replacement for both of those tools called Piximi, and it's just getting started, so it's really in very alpha form at this time, but it uses deep learning in the background, and it has that kind of interface of allowing biologists to label example phenotypes in their image sets. So I hope if anybody is interested in working on this, we're having a hackathon coming soon, and I'd love to tell you more about it. So those are all the tools that I wanted to describe that involve just measuring what biologists want to measure. So those are the known phenotypes. So now let's turn to using images in a more data-driven way. And so what do I mean by profiling? Image-based profiling is this strategy, which I didn't invent. It's been something that people have done for a while, but it's really starting to take root, and really it's starting to become clear that there's some really powerful things that we can do with it. So image-based profiling is when you use images to create signatures of genes or compounds or diseases. So you have some images of cells that have been stained in some fashion, and the appearance of the cells tells you something about the perturbation. And again, those perturbations could be drugs, they could be CRISPR or overexpression of genes, or they could be different patient cell lines. So however you've perturbed your biological system, you take some images, and then you let the data extracted from those images tell you what's unique and different about the different samples, so that you can map, for example, similarities or connections or dissimilarities across all the samples in your experiment. So I will make this more concrete with some actual examples of why you would want to do this and what kinds of things can come of it. But first I just want to say, well, if you don't know if you're not measuring a particular phenotype, how do you stain this? What do you stain the cells for if you don't know what you're looking for? And our answer to that question, well, the short answer is stain them for whatever you want, whatever you think might be relevant to the biological questions that you're asking. But we attempted to build this assay called the cell painting assay. It's not a magical assay. It's really just let's find the fluorescent dyes that are robust and useful and inexpensive, and combine them and stuff as many of them as we can into a pretty simple image-based assay. So there's a Nature Protocols paper that makes it pretty easy to troubleshoot this in your lab. This doesn't have to be in a high-throughput experiment. You can just buy the dyes, add them to your cells, and get lovely pictures where you're labeling a lot of the different organelles within the cell. So this is one assay that could be used for image-based profiling, but I don't want you to get hung up on that. I really want you to think of, in general, any images that you have. You can process them in this way and look for differences and similarities. Cell painting has been used primarily in cultured cell lines so far, but we have ideas for how we could extend this to tissues. Some researchers have converted it to work well on neurons, and as well co-cultures, which are not exactly tissues in a dish, but they're at least a little more complex than cultured cancer cell lines. Okay, so how can we use this rich information? What is measuring the similarity between images and the differences between images? What does that do for us in practical terms? So I'll start out with an example that I can take no credit for. This is from the University of Utah several years back. And they stained some healthy, just normal cells growing in a dish with a few stains. And then because they were interested in this particular disorder called CCM, they knocked down the gene that is known to have a loss of function monogenic causative role in this CCM disorder. And so when you knock down that gene, the cells look obviously quite different to our eye. This is one of the most dramatic phenotypes that one could see. And then it's a very simple concept to say, well, let's take those cells that are in this disease state and see if we can revert them back to healthy looking. So that's the basic concept of the experiment that they ran. What's really neat about how they decided to do this experiment is they took they allowed the experts in the lab to choose a set of drugs in the screen that reverted the phenotype the best according to their eye, but then they also let an automated analysis choose a set of drugs. And what was really exciting is that the drugs chosen as hits based on the automated analysis outperformed those that the experts picked. So now they scaled this up. They since formed a company, which I'm now involved in, but they have hundreds of disease models created in this very systematic way. So I think this is one of the ways that we can start to start to break Eroom's law and tilt it down a bit. Mikko Taipale is a researcher at University of Toronto, and he has created a really neat system that I think could also be very systematically applied to identifying drugs for particular rare disorders. And it's a similar concept to what I just described. It has a little bit of a tweak to it. Here he's studying rare diseases. There's thousands of them that are collectively not rare, but each individual one is not overly financially incentivized by the pharma system. And so thousands of them are monogenic, and of those, he took about 3,500 variants that were known to cause human disorders. And what he did is he tagged each protein so that you have the localization. So instead of knocking down the protein, as in the previous example, here we're tagging the protein and looking where it's located. And so you see that in the green channel here, the flag tag is where this protein normally is, and you look at it in its wild type form and in its mutant form, and you ask whether there's any difference. And they did this experiment primarily by eye, looking for differences in localization, and found around 100 of them. And then we then got involved in the project and are computationally analyzing those images to see if we can find more localization defects, but also looking at these other channels, which they largely ignored. So in other words, when a particular gene is overexpressed in its mutant form, does it disrupt the structures of any of the other structures of the cell? So that's pretty exciting. If we can identify those changes in high throughput, we can use a method such as the one that Luke talked about this morning, the pooled image-based profiling that he described. You could imagine running an experiment where you take 400 diseases times 5,000 FDA-approved drugs, and in the course of an experiment that takes about a year, you could potentially find quite a lot of therapeutics that would be useful for these different disorders. So that's the highest priority for my lab right now, is running that kind of an experiment. And it's only possibly because of that pooled approach that was described this morning. So that's a pretty high throughput experiment, something that is a bit smaller scale, but a very important one in terms of the potential clinical impact is this collaboration we have with Bruce Cohen and Donna McPhee at McLean Hospital. And so again, the concept is quite similar to what I've described, where you have cells taken from a patient without mental illness and cells from patients that have a particular disorder. You take pictures of their cells with some stains, and then you compare those two, and you ask, is there any difference between class A and class B? So it's a pretty straightforward machine learning problem there. And what I'm showing here, just to kind of demonstrate this visually, I'm not showing the statistics that would actually be convincing, but just to give you a visual of how this kind of experiment works, here we have feature space. So the axes don't mean anything in particular. It's just plotting similar cells near each other. So each dot here is a single cell taken from a patient, and it's a group of maybe 30 patients in each of these classes that's grouped together. So this is what controls look like. You could stare at this plot. It doesn't really tell you anything about anything until you look at the other classes. So if you look at patients with bipolar disorder, you see that there's this region of the plot here that's a little sparse, and that's easier to see if you overlay the two. And then you can see this region of the plot is pretty blue. We see a similar thing for schizoaffective. We don't have as many patients, so there's fewer cells overall, but we see a similar type of pattern. And then for schizophrenia, we don't see a depletion here, but it's pretty smooth and even, whereas in the controls it's enriched here. So it's definitely a difference overall. Schizophrenia is a disorder that's quite related to bipolar. And then finally, depression, which we're really excited is to see that this enrichment comes back in depression. And we also started to notice on the right-hand side, there's a depletion here that you see in depression, which is a bit actually enriched in some of these others as compared to controls. So again, this doesn't prove anything, but the statistics behind it have made us convinced that these are some phenotypes that are worth screening for. So now we can take patient cell lines from patients that have these disorders and screen for drugs to try to reverse this phenotype that we see in a dish. Another related kind of experiment in this domain involves dynamics. So these are Tim Becker in the lab coordinated some efforts to create data sets where we've got cells taken from patients with asthma, various kinds of asthma. And it's a pretty murky disease overall. It's actually, there's probably multiple varieties of it, but it's a little unclear what's what, and it's unclear who's going to respond to which kinds of drugs. And so the idea was, can we take cells from patients, grow them in a culture system where there's a chemical gradient across the culture so that these cells are known to move towards a chemoattractant. And it depends what the surface is of the cell, it depends what they're moving towards, but we can measure all the dynamic properties of these cells and see whether there's anything that corresponds to different groups of patients with asthma. Can we group those kinds of patients? So I wanted to mention this project simply because it had a kind of a neat finding in terms of how to combine morphology and migration information. So once you add dynamics to an experiment, you just have a tremendous explosion of features that one could measure. So if we take all those dynamic features and combine them with the baseline morphology of the cells in a more snapshot kind of way, the simple thing to do is just to concatenate those features and let the machine learning algorithms sort out which ones are important. We actually found much better results if we first created a similarity matrix and then used graph-based fusion. And the results of that are shown here, the concatenation versus the fused method. But I thought that was a nice way to combine the data that allows each of those data types to provide some input into the final answers and into how to classify these patients. Another way we could help the drug discovery process accelerate is assessing the mechanism of potential drugs based on their profiles. And so in this kind of a designed experiment, you take a compound that, if you're a drug company or a biological laboratory that has a compound that has some really interesting property in some biological system, but you want to know more about the mechanism. What's the target of this drug, or which pathways is it impinging upon? It's a pretty challenging step in the drug discovery pipeline, and a lot of projects get bottlenecked here in pharma. And so if we have this query compound, one thing we can do is we can treat cells with it, take their pictures, and extract these profiles, and then look them up against a library, a lookup table of existing chemical or genetic perturbations, and see if they match. So this strategy has been adopted for a few years in several pharma companies where they use this kind of routinely to look up drug activities. But I wanted to show a piece of data that is new and unpublished and pretty exciting for those of you who everyone in this room, I'm guessing, loves images and thinks they're a great data source. But you may come across people who are coming from a more molecular mRNA transcriptional world where they think that transcriptional profiles are everything, and imaging is just sort of a poor proxy for some features. So you're about to be very happy with the data I'm going to show, because in this experiment, what we did is we took small molecules, a few thousand compounds, and they fit into 155 categories. And those categories, you have to understand, are a little bit mushy. They're categories that define the mechanism of action. So it would be things like an EGFR agonist or a microtubule inhibitor. So these are categories that have where our humanness, it's probably flawed, but we have some sense of how these drugs work. And so taking those compounds and running them through the cell painting assay, extracting their features, and then we computed a correlation matrix, which I've ordered in a funny way here so that you can see the data a bit better. So each row and column here is one of the 155 categories. And if you see something on the diagonal, it means that the compounds that are in that category are highly correlated with each other. And so having a red diagonal is a good thing. And what I've done is I've reordered the rows and columns so that all the data is pushed up to the top left, so you can just see it a little bit better. And then I've done the same thing with a separate kind of data. Instead of imaging the cells, they were ground up and lysed, and their mRNA profiles were taken using a technology that is high throughput called L1000. This is the only one that's really practical to do in a large-scale kind of way. It's still about 5 times more expensive than an imaging assay, but it's the best you can do with the constraints that are anywhere close to imaging. And so what was really surprising even to me is that we see much less of a signal on the diagonal here with the transcriptional profiles. I think myself, with my own training being as a cell biologist, I've always loved morphology and cell structure, but I would not have predicted that 6 stains of cells would have as much information as 1,000 mRNA levels from a cell. So we haven't dug into this experiment too deeply yet to understand things like what are the sort of off-diagonal metrics. I think in many cases these categories sort of overlap. For example, one might be targeting one protein, the other might be targeting another protein, and labeled as separate classes, yet they might be impinging on the same pathway. And so maybe some of these diagonal signals might make sense. Some of them might be technical artifacts. So we still are digging into it to learn more, but I think this is pretty encouraging and pretty exciting for the power of images to yield very specific information about how the cell is experiencing its environment. When we have mRNA and transcriptional data side by side, we can do some really neat things. And so the Broad is part of a huge effort known as the Human Cell Atlas. And my lab specifically is not terribly involved in it yet. It largely focuses on mRNA readouts at this time. But we were kind of looking ahead and thinking, well, it's great if we have an mRNA map of every cell type in the human body, but how do we correlate the mRNA map to what pathologists know of different cell types? You can do it in a kind of ad hoc, one at a time way based on existing knowledge of which biomarkers might be present in which cell types and so on. But maybe there's a systematic way that we can link these individual separate maps. If we have an mRNA map of cell types and a protein map of cell types and a pathologist assessment of cell types, is there any way that we can map all these together? And so this led us to propose the Rosetta project, which we're currently in progress. I don't have any results to show specifically, but the goal of the project, I think, is a neat one to ponder. That is that can we learn a function where the morphological profile, if you give me a morphological profile, I can give you the transcriptional profile of the corresponding cell, and vice versa. If you give me a transcriptional profile, I can give you a morphological profile back. And the way we're doing this is using data where we have both data types available, both the morphology profiling by cell painting and the transcriptional profiling by L1000. One of the examples is the one that I just previously showed you with the different drug classes. And can we learn these functions from this data? What's neat about this and about the fact that generative modeling is a way that you can extract features and create models. What's really cool here is we can go beyond just predicting a morphological profile. What we can then do is synthesize images that reflect the change that you would see with a certain transcriptional profile. So in other words, we can synthesize some baseline cell with some baseline properties, and we can say, okay, if I push these genes up and those genes down, what would the morphological impact look like based on all this data that it's learned about all these different perturbations that we've tested? And then synthesize images and be able to really try to wrap our biologist brains around what the cell is responding to. So this is all just a neat concept and a neat idea, but we have yet to see how well this actually works in real life. Another application of image-based profiling is to identify gene and allele functions, and that's by grouping similarity. And this experiment has worked really nicely, where we overexpress, in this case, only a few hundred genes. We do the cell painting and the image analysis to extract all the features, and then we cluster those samples based on similarity. And what we found in this single, very inexpensive image-based assay is that the cell type, the genes that are known to be in the same pathway group together. We find that negative and positive regulators of a pathway give opposite profiles, which is kind of an interesting concept to think of opposite profiles in image-based space. And even in this case led to a discovery of a new negative relationship between two pathways that was previously unknown. So on the basis of these results, we're very excited to scale this up, and just got a grant from the state that will help us to get at least half the genome covered by this methodology. And that data will be made publicly available so that if you have an image-based fingerprint from a query sample that you've created, you'll be able to look it up and see if there's a match in this database, which I think will be really exciting and a very useful resource. That data set will be part of a collaboration where we have 7 pharma companies, each of them contributing a lot of drug data, and the Broad creating a lot of genetic data. So it'll be a nice public resource that we should finish about 3 years from now. So in addition to just looking at genes and looking at natural gene functions, we can also look at alleles. And in this particular example, we actually studied a much larger set of alleles in collaboration with the different labs shown here, Alice Berger and Matthew Meyerson in particular, where they overexpressed alleles that were found in lung cancer, not just for BRAF, but for a whole set. I'm just zooming in on one because it's easier to kind of understand the concept here. And the idea is when you have these alleles that are associated with lung cancer, one of the bottlenecks in personalized medicine is you sequence a person's tumor and you say, okay, great, now we know what mutation your tumor has, but we have no idea what drug to give you because we've never seen this mutation before and we have no idea if it impacts the function of the gene that it's in. Sometimes we don't even know the function of the gene that it's in. So we have no way to tell whether this is the thing that's driving your cancer versus some other variants that we may have found. So this would be a systematic way to solve that problem by overexpressing the wild type version of the gene along with a bunch of alleles that have been found in cancers, imaging and extracting the data, and then making, again, this kind of correlation matrix, and looking at what we see for BRAF, for example, is that the wild type version of the protein is down here with negative control. So in other words, if you overexpress BRAF in this particular cell type under these conditions, the cells don't really care that much. They don't do very much. But if you overexpress a constitutively active version like V600E, you get some other profile, some other morphological change. And what's exciting there is then that we see mutations that are known to not be constitutively active don't look like the ones that are constitutively active. They look like something else. And so one could imagine a future world where if we can make these kinds of maps across maybe many experimental conditions to make them as rich as possible, but if we have this kind of map available and you come in with some mutation that hasn't been seen before, if it maps to this category versus this category, it tells us something about what kind of treatment you might do best with. So that's the ultimate goal of this kind of an experiment. Here, most of the projects I've described that have used image-based profiling have used pretty classical methods of using usually cell profiler to extract the features and then using relatively classical machine learning methods. Of course, we're experimenting with deep learning in this space. We have only this one experiment that indicates that deep learning will be more useful. A lot of times we compare them as sort of head-to-head. This is one case where it has turned out that deep learning has been really useful. And Juan Caicedo now has his own lab, and he's hiring, by the way. He is trained as a computer scientist and is an expert in deep learning. And he had the intuition to train a weakly supervised deep learning model. And so what this means is that you train the model to do something. It's called an auxiliary task. So you train it to do something you don't really care about, but it's a thing that will teach the model biologically meaningful differences to look out for. So the auxiliary task in this particular experiment is let's train a model that can distinguish every single treatment that we have included in the experiment. So in this case, let's train a model that can distinguish every single allele of all these tumors, that can distinguish every single allele from each other. And if you train it to do that, it's a thing you don't really care about, and then you ask, can we find similarities and differences among treatments at the population level, we get much better resolution and much better ability to distinguish individual alleles. And so the intuition is shown here, where in general, when you knock out a certain gene, there's a lot of heterogeneity, don't get me wrong, this is an approximation, but generally the cells should look more similar to each other than they should to something that's been treated with another gene. And again, that's an approximation. Sometimes two genes have the exact same effect on a cell population, but that's enough information for this auxiliary task to kind of learn a space. And so I really like this idea of using deep learning as a feature extractor that we can then do interesting downstream applications with. So that's one example of how that's being used. And here's a picture of how that looks in real life. We have three populations here. Each cell is a dot. And we have the control situation is green. In red is the wild type. And you can see there's definitely some cells in the wild type situation that don't look like controls. And then if we have a mutation, it's clearly not overlapping the wild type version. So that gives you a Venn diagram like this, where the mutant is definitely different than the wild type. And so we would conclude from this experiment, from this one snapshot, that this particular particular mutation in EGFR is an impactful mutation because it alters the function of its gene. And we were able to do this experiment without even knowing what the function of EGFR is. It turns out that one's quite well known, but there are many cancer genes where the function isn't known. There is no assay you could do to figure out if it's working or not. So we like this idea as a general approach. And then the last application that I want to describe is called, is all about predicting the outcome of assays based on images as a source of data. And so the impact here is that you can predict many distinct expensive assays on a huge compound library by just running an image-based assay once for an entire large set. So this is most applicable, of course, for a drug discovery situation, but I think there's many ways that we can make this applicable to our kind of everyday practice of biology. So let me describe the concept of it. So the first step is, I guess step 0 here is develop some assay that has some biologically meaningful outcome that you care about. You could be testing drugs or genetic perturbations. Here, the example is with drugs. And you take the results from a small set of drugs, and then you look at the cell painting features from that same set of drugs, and you try to train a classifier that can predict the outcome of the assay that you care about using the image-based data that you have available. So in the super simplest case, there would be one particular feature of cells in the images that exactly predicts the results of your fancy assay that you've made. In a more likely scenario, of course, it will take multiple features combined together, maybe even multiple imaging assays, but in some way or another, you can kind of train this classifier. So it won't work all the time, but for the fraction of the time where this does produce a nice classifier, you can save yourself the trouble of screening millions of compounds against your fancy assay of interest, and instead just look up potential hits, potential drugs that would be useful from the image-based data. And so that's what's shown here. The actual assay results are what you use to build your classifier, but you can then predict the results for the remaining ones, and then you can cherry pick them and test each individual one in the original assay and see how well it does. So nobody has implemented this, to my knowledge, so far in the pharma world. It requires simply just doing one image-based experiment for all 3 or 4 million compounds that they have present. It's an expensive experiment, but if you think about it, every assay that they're able to eliminate is at least $5 million of screening that they would save. So it's not a trivial thing to do this kind of prediction exercise. So nobody's implemented this, but Janssen carried out a really neat experiment that approximates this. So they didn't have a fancy cell painting assay. They just had a sort of everyday imaging assay that they did. It was only 3 channels. One of them was very specific to a particular target. So it's really more like just having 2 image-based channels. And they carried out this kind of thing, and they found for 37% of the assays that they had ever run at Janssen, for 37% of them, they got a pretty decent model, and for 5% of them, they got a really great model with an AUC greater than 0.9. So they picked a couple of those assays that had a great model and that were still actively pursued by the company, and they did this cherry picking exercise and then tested these drugs. Normally, these drugs would give a hit rate of something like 0.5% or 0.1%. And when they cherry-picked based on this instead of random, they got a 60-fold and then 250-fold higher hit rate. And most importantly, they found drugs that were very different structurally, which is an extremely useful property when you're working with compounds that you eventually want to get into humans. So you can read more about it in this study here. Back in our own lab, we've been doing a variation on this. We don't have thousands of assays that we've run over time. We don't have millions of drugs. But what we do have is a very tiny data set where we have 56 genes that have been perturbed by CRISPR. And for each of those genes, the cancer program at the Broad, led by Paquita Vasquez, has tested those set of 56 genes across these different assays that measure cell health. And so they end up with 70 readouts of cell health across these assays, which I think are about 5 physically separate assays. So we were wondering, can one image-based assay replace a lot of those cell health assays? And as you can see here, if we look at the training is on here and test is here, and 0.5 is what you would expect at random, the majority of these readouts, we can get some kind of decent different, we can get some kind of signal. And some of them are quite good. If you look at above 0.8, there's a fair number of these assays that can be very nicely predicted by images. That's exciting because if you remember back, I described the pharma consortium, and this grant is allowing us to make a huge public data resource of cell painting. We can automatically predict the results of these various cell health readouts using that data right off the bat. And you could imagine any assays that you might have, if it's a very difficult tissue-based assay or a zebrafish assay or something that's really hard to scale up, you could imagine trying to correlate the results of a simpler assay with a more complex one using the data that is present in the images. So it's kind of a complex, data science-y example, but there's many other kinds of ways to use image-based profiling. I won't go into any more details. I'll just point you to some resources here where you can read about the applications in the field, look at the best practices of how these kinds of experiments are done. There's a conference specifically, Cytodata is specifically for image-based profiling, and SBI2 for more the image-based screening community to get together. And I'm just showing a few more examples here. So I hope you enjoyed this little tour from measuring what we know we want to measure to using images as a data source and letting the cells tell us their experience and their response to different perturbations. And with that, I'm really happy to answer questions and also point you to the PDF of these slides, which you can access here. And I'd also love feedback on the talk if you'd like to give it. Thank you very much.
Great talk, Anne. Even greater as you ended exactly at 2:20. That's incredible. So everyone, we're going to be taking questions now. Just please wait until the microphone gets to you.
Hi. I really enjoyed your talk. I was really curious how you, or what strategies you might have to deal with, like when you were talking through the Rosetta project, temporal lag, say between RNA expression and phenotype coming in, or that kind of problem?
Oh, yeah, that's a great question. So I wish we had data at various time points for both the imaging and the transcriptional data. In reality, we have a single time point. Usually that time point is not matched. So we debated this back and forth for every experiment we've ever done where we have both data types. We really debate it. Do we want the same time point so that we can compare them side by side, or do we want the time point that's like best for the modality, which for many people would argue for mRNA, you'd want kind of earlier time points to make sure you're not getting too much feedback loops and such. And for morphology, we would argue, well, we'd rather just wait till as much is happening as possible that we can detect. And so the short answer is we don't know anything concrete about how to make those choices yet, because we just don't have any data sets where we have multiple time points to make those kinds of calls.
Yeah, I have the microphone here.
Right there. Sorry.
Yeah, really enjoyed your talk. I had a question about the comparison of phenotype versus transcriptomic comparison. Was the metric showing that the phenotypes are more predictive of the state of the cell than the transcriptome? Is that how we should read it?
Yep.
So then my question is, if the cells are on a surface, and like Gaudenz was saying, that can change the morphology, it's conceivable that the state space of the cell is confined just because of surface.
Yeah.
And then the transcriptome is more noisy.
Yeah.
So it's not as predictive. There's less correlation.
Yeah. So I think what's really exciting for those kinds of an experiment, like it was thrilling to see that we got roughly twice as many drugs classes on the diagonal for morphology versus transcription, but still it's only a third of the total. And so 2/3 of drugs you don't get a consistent profile when you have multiple drugs in a class, you don't get any information. So it's kind of a false negative. And so how can we bump that up? I think one way is to test the cells in different situations. So this is one cell type at one time point with one set of stains. One could imagine using different time points, as was suggested, using different cell surfaces that they're growing on, using 3D surfaces, using iPSC neurons. There's all kinds of different ways that you could expand that out. And it's just a question of how much more information do you get by adding each of those different dimensions of perturbation. But I think each of them could potentially reveal behaviors of drugs that you just can't get with the ones that we've tested, the one that we've tested already. Yeah. Here, I guess.
I think this is a really nice talk, and kind of like indicate a really nice application case how the image analysis could be used for the real drug kind of like discovery. I have a question here. Is that okay? It sounds like the reason why actually the current cost actually is low enough for the large scale screening is because first actually only kind of like a two-dimensional information essentially is extracted, right? And also it looks like because the actual distribution, especially the intracellular distribution of the information is not really characterized. Instead, okay, the overall shape actually is used. So that's the entire process actually is fast enough and compact enough. So that enables the entire kind of screening be possible.
Well, let me just pause one bit. For intracellular information, some of the things we're measuring are the textures of the different stains and the colocalizations and so on. So we do capture some of that intracellular.
Yeah, but not the very detailed kind of like description.
Sure, yeah. I should say most of these experiments are done with a 20x objective, so it's not the highest resolution you could imagine. It's a trade-off, right? Of course, we could do 40 or 60 or 100, but then we have fewer cells per amount of time, and so it's a bit of a trade-off, yeah.
Yeah, so that's my understanding, and my question actually is that, okay, so since you've already been working in this domain for a long time, and if you really kind of like start to include the 3D information and the intracellular kind of like profiling out there, so how much the cost will increase? Like 3 times or 5 times?
Yeah, that's a great question. So taking an experiment from 2D to 3D doesn't necessarily cost more in terms of the sample preparation, it's just the imaging time. And so, it sort of depends, how does your institution charge that? Do you just have the instrument and it's idle some of the time anyway, so who cares if it takes, maybe 10 times as long to capture 3D stacks? Do you need special reagents to make your system 3D as opposed to just 2D would be another component that would feed into that? I mean, the short answer is I don't know. You said 3D, and then what was the other?
The intracellular content.
Oh, the more
detailed. In general, it's a more detailed.
Yeah,
the
so
high resolution version.
If you used a high resolution microscopy, then it's a little bit more simply linear for just the imaging cost. And so there, I think it would only be, maybe 20% more cost to do this at a higher resolution. We've done one experiment so far where we did higher resolution, and we didn't see much impact on the profiles that we were looking at in that experiment. But it was a pretty kind of quick and dirty experiment, so I wouldn't trust that. So we have, as part of this large-scale pharma consortium, we have a plan over the next year where we will actually capture information at very high resolution and kind of the normal resolution we're currently using, and see how much difference we actually see, how much impact it really makes. So I think we'll have a better answer a year from now.
Okay.
Here, Kauditz.
Thanks a lot. That was great. I've actually more of a comment than a question. I'm actually not surprised at all by the result, following up on Charlene, that you get better groupings in phenotypes than transcriptomics, because morphology is driven by kinase activity and protein degradation. And of course, that's what you not see, right? Mm-hmm. That's the blind spot you have if you just look at these endless transcriptomic exercises these days, right? And so now there, I have a bit of a question also / comment. What do you really expect from this Rosetta project? That function, I do not believe.
So that's
for that very reason, you're not going to find a function that converts transcriptome into morphology.
Yeah, you found the fatal flaw, which is that if you believe that mRNA and morphology capture exactly the same things about the cell, then that project should work very well. If there's not so much overlap, then that experiment that function will not be learnable for many of the perturbations that are in the experiment. So we don't have the results yet, but I think you have good reason to be, now that we have, this was sort of started, those projects were started in a staggered fashion. Now that we know the results from the one, I'm a little more skeptical about what we're going to see with the other. It's a neat concept, and I think that it could work in certain domains, and I think the domains where I think it's worth at least trying are situations where we, for example, have an mRNA profile that we know is associated with a disease. Is there a way to translate that to pathology images so that we can make these decisions in a cheaper and quicker turnaround kind of way? Like, there may be isolated situations where it does work, and those would be interesting to follow up, but I think you have a good reason to be skeptical, yeah.
Hi, that was a great talk. I was wondering, in any of these prediction models where you're using the cell painting assay data to either predict cell assay outputs or transcriptomics, have you looked at the data size requirement needs? How many cells you need to look at in order to have confidence in the prediction output?
So not really, not in any satisfying way. So the cell health experiment, we had only 56 perturbations, which I find astonishing that anything worked with just 50 perturbations to move the phenotype space around. So I would expect that you would generally, so in the case of the Janssen experiment, there it seems like you want at least many thousands of drugs to make a decent model. But then of course it depends. If you have an assay where 5,000 drugs gets you 2 hits, that's not a lot of information. But if it gives you 6 or 10, then it starts to be more useful. So there's a lot of parameters there to consider. But I would say as a first approximation, you probably need thousands of these things to make a decent predictor.
We have a question.
Hi, here. Great talk. I'd like to follow up on the same like mRNA to morphological confirmation. We see a lot of explosion of single cell data. Everything is trying to be based on transcriptomics, but traditionally, if we look into the cancer maps or even cross tissue, mRNA to protein correlations have been very low, 40 to 60%. So now, if I look at that image where we are trying to correlate between the morphological versus the mRNA, we are trying to make a relationship between mRNA and protein. Have you looked into the protein connectivity maps from the mRNA and to then correlate them with the morphological? Because that will make more of a kind of direct association if there is an impact in the function build.
No, that's a great idea. We could, I think the thing that is reasonable that we could do with this particular experiment is maybe filter out the mRNAs that are uncorrelated to protein levels based on those existing data sets. I think that's a great idea.
Thank you.
I have a final question here. Is that it sounds like all the screening has been done here. Actually, looks great. Okay, but it happened at a relatively kind of like large and coarse scale. But from this kind of like drug or compound screening perspective, what would be really needed at the end of the day actually is more than just kind of like a screening result for like 1,500 different compounds. But it should actually at the end of the day should really come to like for example for 10 of them, really pinpoint what will be the difference of the cells correspond to all the compound or drugs out there. So I wonder what are the fine scale information the pharma will really need to look at for the small set of the kind of like a final list in your screen result instead of just a whole list of a correlation or this kind of stuff.
I'm not sure I understand the question. So
I try to ask what are the kind of like detailed features from the drug discovery perspective, what will be really needed out of the high resolution version.
So maybe this will address your question, but tell me if not. So in general, they screen millions of compounds. They find, they rank order a list that has some properties. Then they take that list and they often run some secondary assays to kind of look at the phenotype in a different way and kind of narrow things down. And then their medicinal chemists stare at the structures and say, we don't want to make that compound or like this is going to be messy or this is going to be toxic or something. So they kind of filter it that way. And then toxicity folks get their hands on it and they test all the molecules for toxicity. And so in the end, the final decision of what drug to actually proceed is a combination of a lot of quantitative data and a lot of qualitative inputs as well that sort of move things down the pipeline. And so usually the result from a single imaging-based assay is not going to be the sole determinant of the outcome. There's all kinds of other inputs that once things have made it past an initial filter, there's all kinds of features.
So basically for the subsequent steps, so the imaging assay is not really involved.
For subsequent steps, not really, but I think it should be involved. What I didn't talk about predicting toxicity. Well, predicting cell health is some flavor of this, but every drug that goes through a pharma pipeline has to go through a toxicity assessment. And that can be extremely expensive if it involves animal models. And so if you can use an image-based assay to predict outcomes in rats and dogs, that would be a tremendous cost savings because you could make those decisions earlier in the stage of the pipeline. So that would be one thing that they would really appreciate predicting early on.